Hello, my name is Agnes Daudignon. I am cytogeneticist in oncohematology, and I coordinate the working group of OGM in hematology named the FrOGG. It is not to be demonstrated that OGM have taken a big place in cytogenetics, especially in oncohematology. You can see here some of the many publications that cover all the hematological malignancy landscape, like MDS, ELA, myeloproliferative neoplasm, CLL, and myeloma. So we can say that OGM represents the next generation of cytogenetics in the same way as NGS revolutions molecular biology. Since 3-4 years ago, OGM started to develop in research projects in a few laboratories in France. Then, in light of the initial results, the technology rapidly spread. The Groupe francophone de cytogénétique hématologique, the GFCH, decided to supervise and to organize the deployment of OGM in France. This group, the GFCH, is composed of around 100 members of cytogeneticists from French-speaking countries like Algeria, Belgium, Canada, Lebanon, Luxembourg, Morocco, Switzerland, and Tunisia. So the GFCH created the FrOGG group for OGM use, which now include labs from France, Belgium, Switzerland, and Quebec. This group started in January 2023. Until now, the FrOGG is composed of 53 biologists and engineers from 29 labs of hematological cytogenetics. Of note, some of other labs, like in Belgium, are not part of the FrOGG, but rather belong to the Northern European Consortium or both. In this presentation, I will only talk about the labs that have joined the FrOGG. In France, there are 14 labs that are equipped with Saphir for hematological use. We hope that there will be more. So to organize the FrOGG, we subdivided it in 6 working groups, each led by 2 biologists. Each group worked independently. Once or twice a year, the whole group gathers. And there is a constant communication between the subgroups, the FrOGG's members, and the GFCH office. All the information and documents are available on our website for all members of the GFCH. I will now present the 6th group. The first group works on the validation of the method. It is conducted by Valentin Lestringent from CHU of Amiens and Nathalie Gachard from CHU of Limoges. The goals are to define the standard scope of French accreditation and to propose a common solution for the French quality label called COFRAC. This leads to propose a framework to validate OGM in all hematological labs. The second group works on myeloid pathologies and acute leukemia. It is conducted by Nacera Abermil from Hôpital Saint-Antoine APHP in Paris and Hélène Guermouche from CHU of Lille. The goals are to discuss technical aspects, OGM data analysis and bed files, and to organize the reports for the clinicians with the big question, which SV or CNV should be reported. There is also time dedicated for case presentation. The third group works on lymphoid pathologies and myeloma. The goals are identical to the previous group, with some specificity due to the sample, like fresh or frozen biopsy for non-Hodgkin lymphoma, as well as bone marrow or blood if infiltrated, like sorted plasma cell for myeloma, or like blood or bone marrow samples collected before or after cell culture for CLL. This group is led by Christine Lefebvre from CHU of Grenoble and myself. The fourth group is led by Baptiste Gaillard and Faten Soumi from CHU of Reims for the first and from Saint-Antoine Hospital for the second one. The goals are to organize external quality control for OGM data in hematology. First test is programmed on first semester 2024 between labs from this group. In addition, inter-laboratory quality control are organized by sharing patient sample between 2 or 3 labs in order to test the entire process from the sample receipt to result report. We are also interested to participate to an European EEQ. We are for that in contact with Blanca Espinet or Katrina Rack with their respective consortiums. The group number 5 works on the financial aspect and is led by Dominique Pinter from the Centre Anticancéreux from Rouen and myself. The objective is to establish a funding plan to equip all the 41 French hematological labs with OGM. For that, we work in close collaboration with Bionano to submit a special demand named Forfait Innovation to French authority linked to Health Ministry. The purpose is to implement OGM as a clinical tool in all hematological labs in France. Finally, the last group worked on chromoanagenesis. This group is led by Jean-Baptiste Gaillard and by Hélène Guermouche, Jean-Baptiste Gaillard from CHU of Montpellier and Hélène Guermouche from CHU of Lille. All users of OGM have observed that complex events are well highlighted and characterized by the OGM. The goal of this group is to establish an OGM description of the events called chromoanagenesis, like chromotypsis, chromoplexy, and chromoanasynthesis. We are also participating in the creation of a similar group at the European level. So I have presented the 6th group, and now I will present the application of OGM in hematology from the different labs listed in the FrOGG. First, in acute lymphoblastic leukemia. In France, OGM is used in routine in 5 labs and is in progress for the other 9 labs. In Belgium, OGM is routinely used in adult and pediatric ELL in Leuven. In Switzerland, OGM is implemented for all pediatric and adult ALL in Lausanne and is in optimization in progress in Geneva. Of note, in pediatric ELL, OGM has been accepted as an alternative to FISH, RT-MLPA, and DNA array in the International European Protocol altogether. Now, for EML, OGM is used systematically for all cases in Lausanne, Switzerland, and Leuven, Belgium. OGM is in optimization for the same use in Geneva and Montreal. In France, it is used as a complementary technique for now in 7 labs. For myelodysplastic neoplasm, the position of OGM is still being discussed. However, OGM is used as a replacement to karyotype and FISH in one French site, Necker Hospital from Paris, as a complementary technique at Brest Hospital in France, in some cases with high blast level in Leuven in Belgium. Optimization is in progress in Geneva in Switzerland. For myeloproliferative neoplasm, OGM is not implemented in routine. We have a clinical research project in progress in the north of France, led by Valentin Lestringent for Amiens. This study on the 300 patients whose objective is to compare OGM with current cytogenetics, karyotype and FISH, and to evaluate the informativity of OGM in these indications. Another study is conducted in Lille in collaboration with the CEREO, a network of hypereosinophilia in France, to assess OGM impact in the detection of anomaly in this pathology. In Belgium, OGM is being validated for HS2 for routine implementation. For multiple myeloma, in France, we are publishing a paper for the feasibility of OGM in myeloma. OGM will be implemented in routine in CHU of Reims and in CHU of Versailles and Rouen as soon as they are equipped. In Switzerland and Belgium, OGM is being tested In Quebec, a paper was published and routine use is planned. Finally, the application in non-Hodgkin lymphoma has been investigated by 3 labs in France and 1 lab in Leuven, Belgium. Christine Lefebvre from the CHU of Grenoble will present her study results at the Congress of French Society of Hematology in Paris on March. For CLL, a retrospective evaluation of OGM is conducted in Saint-Antoine Hospital and La Pitié-Salpêtrière in Paris in the context of GFCH research progress. This was a summary of how the group is organized, how we work, and what is our plan to implement OGM in the labs in France. I want to thank all my colleagues from the FrOGG who work very actively in the subgroups and our colleagues of the GFCH. I want also to thank the Bionano team for their support. Thank you everyone for your attention.